So let us quickly look at the glycan diversity in trypanosomes. As I have discussed so far, that trypanosomes express a diverse range of glycans, be it N-glycans or GPI-anchored glycans. In Trypanosoma brucei, it is known that UDP galactose and UDP gluconic dependent glycosylation pathways are essential, and thus they play a major role in the survival of the parasites. Now, in N-glycans, there are oligomanostructures, quasi-manostructures, and complex N-glycans. There are GPI-anchored glycans, and recently an O-glycosylation is also reported in VSG glycoproteins, but the entire trisaccharide structure of that O-glycan is yet to be characterized. The glycans are synthesized by the action of several glycosyl transferases. The glycosidic linkages are defined by configuration and anumericity of the transferred sugar. Each glycosidic linkage is catalyzed by a unique glycosyl transferase using a specific activated nucleotide sugar donor. In Trypanosoma brucei, there are around 38 unique glycosyl transferases that are predicted to account for 38 known glycosidic linkages. And now you can see here that every linkage is being given a number which indicates towards a glycosyl transferase activity. The glycans here can be alpha gal, beta gal, or beta glucanat. And out of the 38 predicted glycosyl transferases, only 10 GTs are characterized in Trypanosoma brucei so far, and there are other 19 putative UDP gal or UDP glucanic dependent beta glycosyl transferases which are yet to be characterized. Now, some GTs can be stage specific, means they are either working only in bloodstream form or in procyclic form, but there are other GTs which can work in both life stages. We have recently characterized a glycosyl transferase in Trypanosoma brucei which is called TBGT10, and I'm quickly going to describe the method that we use to characterize this glycosyl transferase activity. So this project was led by one of my colleagues. He generated the conditional knockout of TBGT10 in bloodstream form parasites. We noticed that the ricin-lectin binding was highly reduced in the TBGT10 knockout cells. Ricin-lectin especially bind to the beta-galactose residues. And we also employed another antibody called MAP139, which basically binds very specifically to polylacnic repeats. Polylacnic repeats are galactose and glucanac repeats. And we noticed that there was a great reduction in the binding of MAP139 in the TBGT10 knockout cell lines. So the overall reduction in MAP139 and in lectin binding indicated towards the alteration in the total N-glycans. So this would let us think what could be happening to the total N-glycans. And this question for me was to address that what alterations could TBGT10 be doing to the glycans. So the objective was to analyze the total N-glycans. Now there are several already established methods to get the purified N-glycans, but majority of the methods require a lot of material and a tedious process involving enrichment, desalting, deglycosylation, and cleanup. Therefore, we decided to re-establish one simple method that is quick and does not need any enrichment or deglycosylation. So I worked upon this method establishment and managed to optimize it for N-glycosylation. What all I need to do is just prepare the cell lysate that contains the total glycoproteins and digest it with pronase enzyme. Now pronase enzyme is a nasty enzyme because it chops up everything, but it is very efficient here because pronase digests the entire protein into single amino acids, and then one amino acid residue would carry the glycan. Then just clean up the digest with chloroform phase extraction to get rid of all the hydrophobic contaminants, and the glycopeptides are in the aqueous phase and ready to analyze on the GCMS. So once we have the glycans or glycopeptides ready, we can do two things with GCMS. First is analyzing the total monosaccharide composition, which would tell us what sort of sugars or glycans are there in our sample, be it like galactose or mannose or glucose or glucanac or galnic. Second is to analyze the glycosidic linkages. And it is a little bit complicated process, but very efficient to identify the glycosidic linkages. So for monosaccharide composition, we do a process called methanolysis, which is basically a hydrolysis process, and it hydrolyzes the glycosidic linkages, and then we finally get the methyl glycoside of monosaccharides, and then we analyze these methyl glycosides of monosaccharides on GCMS. Now based on our standard sugar standard, we know the retention time of each sugar, and that's how we identify what sort of sugars are there in the sample. So for example, here I'm showing my sugar standard. So it is a mix of different sugars, and we know that what is the particular specific retention time for mannose or galactose or any glucanac or galnic. And we use the silo-inositol as our internal standard. And based on this profile, we can identify what sort of sugars are there in the samples. And the next process is to do the sugar linkage analysis, and we call it PMAS preparation, which is partially permethylated aldol acetates. And it is a complicated process a little bit, but involves four chemical modification. First is the permethylation, where we permethylate all the free hydroxyl groups, and then we hydrolyze the glycosidic linkages with acid hydrolysis, and then we do the reduction with sodium borodeutride, which enables us to distinguish between C1 and C6 carbon. And then we acetylate the newly generated free hydroxyl group, and that's how we get the final PMAs of the sugars. Now, so for example, here we have 1,4 linked sugar and then this 1,3 linked sugar. So 1,3 linked sugar in the blue basically gets the acetyl group on the C3 position, and the red one, which has the C4 involved in the glycosidic linkages, get the acetyl group at the C4 position. And that's how we identify the glycosidic linkages and how the sugars are linked to each other.